Hi everybody, today we are going to talk about variations of photosynthesis. So what we're really going to focus on today is what happens when some of the aspects of photosynthesis go wrong. So that is what this means. We're really going to be looking a lot at some evolutionary concepts. So we're going to be looking at evolution. So be on the lookout for that. Think, kind of remind yourself now, okay, what is evolution? Evolution is when things with beneficial adaptations survive and carry on with offspring, and things with not so beneficial adaptations don't survive and they don't carry on their adaptations through offspring. They're less likely to. So that's what we're going to talk about today. So a few reminders before we begin. Number one, remember what plants need. Plants need, in order to do the light reactions, plants need a few things. Light. Remember, light energy is entering the plant, it's hitting the plant at the leaf. So that comes from the, and then we know water is coming from the roots, it's coming from the ground. And we already learned about that, how osmosis pulls the water in, and that negative pressure pulls the water up. And then, of course, there's the Calvin cycle. The Calvin cycle requires carbon dioxide, which comes from the air. So what structures have plants evolved to supply these needs? Well, let's think about that, right? We know a few things about some adaptations. Number one, leaves evolved to be flat and broad. And although not every plant has flat and broad leaves, many do, and that's to collect as much light as possible. Plants have evolved roots that absorb water with a high surface area. That's why roots have root hairs, to absorb as much water as possible. And then there's CO2. So there's the root. The CO2 enters through the leaf as well. So here is the leaf structure. So imagine this is like the thickness of a leaf. So right there, like not the broad part, but the thickness. And there's a palisade layer, which has all these column-like cells. These are good at absorbing light. And then we have the spongy layer that's good at keeping air. Epidermis and a waxy cuticle that keeps water in. And then we have the stoma, which I'm just going to refer to as the stomata. The stomata are openings that let CO2 in, or they can close and they can prevent water vapor from leaving. So two things happen with the stomata. Remember, the stomata open, but when they're open, CO2 can come in, but also water vapor can leave. And then they can close. And when they close, that might prevent water vapor from leaving. It will also prevent CO2 from entering. The guard cells, these are the cells that actually close and open the stomata. So we see water vapor can leave. That is usually fine unless you're in a drought, and then that is not a good thing. It also allows oxygen to leave, which is generally a good thing because plants need to get rid of that waste product. And it also lets CO2 in. So we have gas exchange happening through the stomata. It's kind of like the lungs of the plant. Then we have xylem. The xylem, remember, just bring up water. We also have phloem. The phloem is for the sugar produced during photosynthesis. Okay, so how can we control water loss from leaves? Basically, plants used to exist only in water, and then eventually evolution drove plants to growing on dry land. Now, the stomata evolved in response to that, right? The stomata prevent too much water from leaving the plant. So they are a response from evolution of land plants. And on hot or dry days, the stomata are going to close. And when they are closed, we see this down here. That is a closed stomata. It prevents water vapor from leaving. All right, those guard cells are going to close or open. So if the guard cells are full of water, so if they are in a hypotonic solution, water is going to enter the guard cells, kind of like those floaties that kids might wear. They're going to be open. And if they lose water, they're going to wilt and they're going to shrivel together and they're going to close. So this is an adaptation to living on land, but this is to create problems. What are the problems? Stomata leads to water vapor, so it saves water, but it prevents CO2 entering. It also creates an O2 buildup. Remember, the light reactions are producing O2, and if the stomata are closed, the O2 is going to build up. It's also going to lead to a CO2 depletion because new CO2 cannot enter the plant. So, two unfortunate things happening. We might be saving water, but we have too much O2 and not enough CO2. So this is going to cause problems in the Calvin cycle, as our penguin is reminding us. All right, so what is going on? What is exactly the problem? The problem has to do with rubisco. Rubisco, remember, is that enzyme that fixes carbon. There's a diagram of rubisco. Rubisco is the carbon fixation enzyme, and it normally bonds carbon from CO2 to that 5-carbon RUBP. Remember, in the Calvin cycle, 5-carbon RUBP got an extra carbon from CO2. CO2 is the optimal substrate, and it's usually going to cause the reduction of our sugar, and it's usually going to cause building, right? Because a reduction usually refers to building. And that's normally how photosynthesis goes. Unfortunately, when the stomata are closed and the O2 concentration gets too high, the rubisco can actually bond oxygen to RUBP instead of carbon. So O2 is going to be a competitive substrate, basically, right? Remember competitive inhibitors? So if there is too much O2 and not enough CO2, O2 is going to get fixed and not CO2. This is going to lead to oxidation. Oxidation is the opposite of reduction. So we're talking about the breakdown of the sugar and not the building of the sugar. So this is called a let me move myself so you can see that term, photorespiration. Photorespiration is not a good thing. So normally, when CO2 is abundant, this picture is just showing us the Calvin cycle. The Calvin cycle happens and we build sugar, and that's exactly what should happen. This goes carbon and we make glucose. This is what should happen. This normally happens with what we consider normal plants. These are called C3 plants, and most of the plants you would find around you are C3 plants. All right. C3 plants usually undergo normal Calvin cycle because there's plenty of water. So there's not these long periods of drought where a plant is going to have to shut down and it won't undergo photosynthesis as well as it should. However, this is a problem if you live in a hot and dry climate. You are much more likely to undergo photorespiration. When O2 is high, so if you're a plant in a hot and dry climate, O2 is going to bind, and instead of making 2 3-carbon compounds, one of those compounds is just going to be a 2-carbon compound. And that 2-carbon compound is basically going to go to the mitochondria, and it's going to get lost. And it's not even going to make any ATP because it can't. It's not quite the right compound. So we're just losing about half of our sugary compounds through photorespiration. So rubisco is not doing us any favors if the O2 concentration is high. So the impact, basically we have oxidation of RUBP, and that is going to short circuit the Calvin cycle. So this is going to be a loss of carbons as carbon dioxide, and this can be up to a 50% loss. So that is a huge loss. Plants in a hot, dry climate are not going to do as well using traditional photosynthesis. So it reduces the production of photosynthesis. No ATP is produced. No glucose is going to be produced, or a lot less glucose is going to be produced. But if we could reduce photorespiration, if we could lower it as much as possible, the plant is going to become a lot more efficient. So in hot, dry climates, there have been selective pressures So there have been pressures that evolved alternative carbon fixation systems. So there are plants that exist in hot, dry climates, and they can deal with photorespiration a little bit better. So we have C4 plants. C4 plants are going to physically separate carbon fixation from the Calvin cycle, and we'll find out what that looks like. All right, so C4 plants are going to use different cells, so basically different locations. That's going to help reduce photorespiration. All right, and they're also going to use a different enzyme called carboxylase. We're not really going to spend a ton of time on the details in this unit. C4 plants also have a slightly different leaf structure because they have these different cells that allow for physical separation. And then there's CAM plants. CAM plants to separate carbon fixation from the Calvin cycle by time of day. All right, they're going to fix the carbon at night and then store the carbon that is fixed until the daytime because the stomata are going to be closed during the day to conserve water. So the carbon has to be fixed and stored until photosynthesis starts happening in the daytime when there's light, and then it's going to continue on. The stomata open at night. And there's a CAM plant. So let's start with the C4 plant. This is a better way for certain plants like corn or sugarcane to capture CO2. The first step before the Calvin cycle, carbon from CO2 has to be fixed, and it's basically going to get fixed twice. And this will keep O2 away from RUBP. It will prevent photorespiration. That's a key point. So it's going to store the carbon, and then it's going to move it to a different cell. This is an adaptation to a hot, dry climate where the stomata have to be closed often. So leaf anatomy is different. Sugarcane, corn, certain grasses have this. Look at what's going on. Okay. We have a different cell called the bundle cell that is present. And basically CO2 is getting fixed into a 4-carbon compound. And that 4-carbon compound is going to move into a new cell. So photosynthesis is happening up here, but the carbon fixation is going to go and it's going to move the carbon into a new cell where we don't have as much oxygen because all the oxygen is happening up here. So the 4-carbon compound is then going to move into the bundle sheath cell where CO2 is going to be released and then the Calvin cycle is going to happen as normal. So it's a way of keeping the oxygen up here to prevent photorespiration in this other cell. A new enzyme, PEP carboxylase. PEP carboxylase is not going to be very attractive to CO2. And then it's going to regenerate CO2 in those inner cells, keeping oxygen away from the Rubisco enzyme. All right. Here the anatomy, not too much to see here. We've got these huge bundle sheath cells. And then we have CAM plants. Plants are an adaptation to a very hot, dry climate, and they separate through timing. So the stomata are closed during the day. This prevents water loss. And the stomata then are open at night when water is less likely to. So at night, the stomata are going to fix the carbon into 4-carbon storage compounds. And during the day, those 4-carbon storage compounds will release CO2 and the Calvin cycle will happen as normal. So this is going to increase the concentration of CO2 in the cells and prevent competition from oxygen. So succulents, cacti, pineapple, they are CAM plants. And CAM plants are all about timing. Here are examples of CAM plants. And here is what is going to be happening in a CAM plant. So a CAM plant is going to fix CO2 only at night, and then the stomata can close during the day, but there's CO2 being released during the day. It's already there, present as this organic acid. And so the CO2 can drive the Calvin cycle to make sugar, even though the stomata are closed. The C4 plant fixing it in another location, CAM plant fixing it based on time of day. So these alternative methods can be kind of tricky. If you have any questions, let me know. Here's the last part of the notes, just kind of what I said. Okay, so why do we even have this problem? Why do we have the C3 plant? This is a normal plant. Why do we have this problem? It's because when Rubisco evolved, it evolved when there was a ton of carbon dioxide in the atmosphere. And now there's a lot less carbon dioxide in the atmosphere and there's a lot more oxygen. So this isn't quite how Rubisco evolved. So there was not a strong selective advantage if it accidentally fixed a little bit of oxygen. It wasn't that big of a deal because there wasn't that much oxygen back then. Today, the atmosphere is different. There's way more oxygen than CO2, and we have this problem. But solution is working and has worked already to fix this problem. Right, so that is it for some variations on photosynthesis. That's pretty much it for photosynthesis, so let me know if you have any questions.